We talk a lot about pushing the boundaries of nanoscale printing in your day-to-day workflow, like tuning photo initiators, dialing in those slicing
Yeah,
parameters.
the usual daily grind.
Yeah, exactly. But I want you to step back for a second. Picture the ultimate application dream here.
Hmm.
Imagine being able to achieve high-throughput, high-resolution 3D organ mapping, right down to the cellular level.
Oh, wow. Yeah, that's the holy grail.
It is. We are talking about spatial proteomics, unlocking the exact molecular makeup of tumor microenvironments. No guesswork, no blurred signals from mixing hundreds of cells together into some kind of biological smoothie.
Right. You want the precise map of the neighborhood.
Exactly. That is the dream. And welcome to this deep dive. Since you are an expert in two-photon polymerization, or 2PP, we are tailoring today's analysis specifically for you.
Yeah, and it is the ultimate frontier in pathology right now,
Right.
because understanding cancer heterogeneity means we can't just look at what proteins exist inside a tumor. We have to know exactly where those proteins are situated, literally cell by cell, because a tumor is an ecosystem. Some cells are driving the growth, others are suppressing the immune system, and you only figure out which is which by isolating them spatially.
Which brings us to the core of what we're unpacking today. We're looking at a groundbreaking new paper from researchers at Stanford University and the Pacific Northwest National Laboratory.
And this paper is wild.
It really is. They used 2PP to engineer a multi-tiered microscopic blade array. They call it a dicer. And it's designed to mechanically chop human cancer tissues into perfect 10-micrometer cubes.
10 micrometers, that's just incredibly small.
Right. And they did it to completely preserve the protein content inside those tiny cubes.
Right.
So, okay, let's unpack this. You know how to print at the nanoscale, but today we are exploring how your field is uniquely positioned to solve a massive, seemingly biology-centric problem that traditional lasers simply cannot fix.
Yeah, because before we even analyze the 3D printed geometry of this dicer, we really need to examine the biological bottleneck it's built to shatter.
The dreaded 10-micron wall.
Exactly. In this field of spatial proteomics, scientists have to physically isolate these tiny regions of interest. I mean, you can't just put a whole chunk of squamous cell carcinoma tissue into a mass spectrometer and expect to find out what is happening in one very specific microenvironment.
Right, it would just be noise. You have to extract that specific 10-micrometer neighborhood first.
Yes. And biologists have traditionally done this with a laser capture microdissection, or LCM.
I mean, it sounds incredibly high-tech, like very precise.
And, well, at larger scales, it is highly effective. The standard LCM workflow involves mounting a tissue slice onto a specialized polymer membrane. Usually it's a polyethylene naphthalate, or PEN membrane.
Okay, a PE membrane. Why that specific material?
It's chosen because it absorbs UV light. So the scientist uses a UV laser to trace a path, cutting through both the tissue and the membrane at the same time. Then a laser pulse literally catapults that micro chunk of tissue into a collection tube.
Wait, catapults it. I love the visual of a microscopic laser trebuchet.
It's basically exactly that.
But, okay, if it works well enough to be the gold standard, why are the Stanford researchers trying to replace it with 3D printed blades?
Well, the operative phrase there is at larger scales. When your region of interest shrinks down to the cellular scale around 10 micrometers, the physics of LCM just turn violently against you.
Because of the heat.
Yeah, exactly. A 10 micrometer microtissue has a tiny absolute protein mass, right? But it has a massive surface to volume ratio. So when the UV laser interacts with the sample, the photothermal heat doesn't just cut the edge, it spreads.
Oh, I see. So at 100 micrometers, you're just slightly cooking the outer boundary of the sample, but the center survives totally fine.
Precisely. But at 10 micrometers, the boundary basically is the center.
Yikes. So it just burns it.
The heat literally vaporizes the target tissue along the cutting path. The confocal imaging data from the study is, well, it illustrates this failure pretty brutally.
What do the images look like?
The 10 micrometer microtissues generated by LCM retain only about 25% of their expected volume. The catapulting process literally blows cavities straight through the microtissue and the PEN membrane. The researchers diplomatically call them through holes, but it's basically a crater.
Wow. So using LCM at this scale is basically like trying to surgically slice a single grape with a blowtorch while the grape is glued to a piece of tape.
That is a horribly accurate analogy.
Speaking of tape, what about that PEN polymer membrane? I imagine a melted plastic sheet doesn't play nicely with delicate proteins.
Oh, it's a nightmare. The membrane introduces catastrophic problems for the downstream proteomics. It's highly hydrophobic, so the surviving tissue adheres to it stubbornly, which completely hinders the chemical buffer penetration you need later to actually extract the proteins.
So you're left with a tiny fraction of charred tissue superglued to a plastic sheet that actively repels your extraction chemicals.
Exactly, it's useless.
Well, if the laser is the problem, I'd assume the obvious alternative is just a mechanical knife.
Yeah.
I mean, don't biologists have automated scalpels or tissue choppers for this exact reason?
They do, and the researchers actually ran that exact baseline comparison. They used a standard commercial McLemoyne tissue chopper equipped with a stainless steel razor blade.
Yeah.
The results were an absolute disaster.
Let me guess. A macroscopic stainless steel blade trying to make 10 micrometer cuts on squishy tissue is like trying to slice a tomato with a baseball bat.
Yeah.
Just crushes it.
You nailed it. The friction is the real killer there. Because a macroscopic blade has to apply normal force across the entire tissue slice all at once, it generates excessive friction and heat.
Which means it drags the tissue.
Right. That leads to totally inaccurate blade displacement. A massive portion of the tissue remained entirely uncut, and the pieces that did shear were just scattered everywhere because of the dragging motion and the poor adhesion to the glass slide. They couldn't even generate microtissues smaller than 100 micrometers, let alone 10.
Okay, so we have a clear engineering gap here. Lasers burn the tissue, and stand-metal blades crush and scatter it.
Which means this requires extreme mechanical precision at a microscopic scale.
And that brings us directly into your wheelhouse as a 2PP expert. The Stanford and PNNL researchers recognized that thermal damage was unacceptable and macroscopic cutting was just too clumsy.
They needed to engineer a microscopic mechanical solution from scratch.
So let's get into the manufacturing specs, because the 2PP setup they used is brilliant. They printed these dicers using IPS photopolymer resin on indium tin oxide, or ITO, coated glass substrates.
A very solid standard choice for this kind of structural work.
Definitely. And they used a 25x objective to hit a target blade tip diameter of just 1 micrometer.
And that 1 micrometer sharpness is the critical threshold here. It's sharp enough to cleanly sever ethanol-fixed squamous cell carcinoma tissue without requiring excessive downward pressure.
Because that tissue is pretty soft, right?
Yeah, it has a relatively soft Young's modulus on the order in about 100 kilopascals.
Now, detail that really jumped out from the source, and something I know our listener will definitely appreciate, is the actual hardware they used. They utilized nanoscribe systems.
Ah, yes, the industry workhorse.
Specifically, the paper mentions using the older generation Photonic Professional GT alongside the newer Quantum X.
That hardware choice directly impacted their prototyping methodology, I'm sure.
Oh, absolutely, because of the print times. To reduce the time in the older nanoscribe system, the researchers actually had to shrink the blade array area significantly just for their initial testing.
Oh, wow. How small did they have to go?
For the 10 micrometer space blades, they only printed an 80 by 80 micrometer grid. Because you know firsthand how slow print times can bottleneck iterative development when you're waiting on a machine.
Oh, I know. But we really need to contextualize this within the broader trajectory of 2PP technology, because the industry is currently riding this incredible 20-year-long exponential trend.
The speed-up trend, right?
Exactly. We are seeing roughly a 2x speed-up in printing capability per year. So this really frames the dicer not as some slow niche curiosity, but as an incredibly scalable, mass-producible solution for the near future.
And that explains why they brought in the newer generation Quantum X. That faster system is totally critical for scaling this technology up. Ultimately, their final design called for 50 by 50 arrays meant to stamp out 2,500 microtissues in a single pass.
2,500, that's amazing. But what's fascinating here is the physics of the cutting process. Because printing the blades is one thing, but designing them to cut without crushing the tissue is an entirely different mechanical challenge.
Wait, what do you mean? You can't just print a miniaturized flat grid of intersecting 1-micron blades and press it into a tumor slice?
Not at all. I mean, you'd think so, right? If the grid is smaller, you're displacing less total tissue. Shouldn't it be easier to push through?
Yeah, that's exactly what I assumed.
But the physics actually invert because of the force scaling problem. Cutting energy scales approximately linearly with the total blade length that's in contact with the tissue.
Oh, Right, because 10 micron spacing means way more blade density.
Exactly. If you want a 10 micrometer grid, your blades are packed incredibly tight. In a given square millimeter, you have 10 times more physical blade material engaging the tissue compared to, say, a 100 micrometer grid.
You're basically pushing a solid wall of resin into the sample.
Yes. Applying 10 times the force to a 10 micrometer thick, ethanol-thick slice of tumor tissue just doesn't cut it. Literally. The tissue plastically deforms, it crushes and tears before it ever reaches the shear threshold required to separate.
Which leads us to the absolute engineering breakthrough of the paper.
Mhm.
This is my favorite part. The researchers totally abandoned the flat grid and introduced a multi-tiered, hierarchical blade architecture.
It's such a clever design.
Isn't it? Instead of hitting the tissue all at once, the blades are staggered in height. It looks like a city skyline of razors.
They designed a very specific 20 micrometer vertical offset between consecutive tiers of blades.
And wait, the squamous cell carcinoma tissue slices they were using were exactly 10 micrometers thick. So that 20 micrometer offset is exactly twice the thickness of the tissue slice.
Yes, and the mechanism here is beautiful in its simplicity. The top tier of the blade engages the tissue and cuts all the way through to the glass slide, which completely frees the edges of that specific microtissue block before the second tier of blades even makes contact with the top of the sample.
Oh, wow. They're dividing the required cutting force across time. You're never fighting the entire grid at once.
Exactly. The researchers backed this up with hard data using an Instron force displacement testing setup. They mounted the 10 micrometer tissue slices on wax-coated glass slides and measured the maximum indentation force.
And how did it perform?
Moving from a flat blade to a serrated blade reduced the maximum force to about 30% of what the flat blades required, just simply due to the stress concentration at the serration tips.
But the multi-tiered design is where the real magic happens at the 10 micrometer scale, right?
Absolutely. At that ultra-tight spacing, a 4-tiered serrated design required only 19% of the cutting force of a 2-tiered design.
That is a massive reduction.
By staggering the blades across 4 distinct levels, they reduced the mechanical stress on the delicate tissue by over 80%.
Which is incredible. But here is where it gets really interesting. Because you might think, great, I have a super sharp 4-tiered 2pp blade array. I can just use it like a biopsy punch all day.
Right, just wash it and reuse it.
Exactly. But the source material exposes a gritty reality of this kind of microengineering. These 2pp blades are incredibly sharp, but they dull almost instantly.
Well, it comes down to the material properties of the IPS photopolymer. It's interacting with dense biomatter and bottoming out against the wax substrate.
Right. The researchers actually ran a test with a single flat blade, performing 2 indentations on different regions of the same tissue slice. And on just the second cut, the maximum cutting force required more than doubled.
The tip was already blunting.
Exactly. So these 3D printed dicers are strictly single-use consumables.
Which is actually a feature, not a bug, in biological applications.
Oh, true, because of cross-contamination.
Exactly. Single-use devices entirely prevent cross-contamination between patient samples. You use the dicer once to ensure a perfect, mathematically precise cut with minimal force, and then you just discard it.
Okay, so the mechanical physics work flawlessly in a force displacement test. The tissue doesn't crush. But the ultimate metric for this deep dive is the biological output. Does this physical 3D printed geometry actually save the proteins better than a high-end laser system?
To find out, the Stanford and PNNL team set up a direct showdown. The dicer versus LCM, evaluated by liquid chromatography mass spectrometry, or LCMS.
The heavyweight championship of spatial proteomics. I assume they had to keep the sample prep as identical as possible to make it a fair fight.
They did. For the dicer, the freshly diced microtissues were pushed through the blade array into 70% ethanol and then transferred to nano-POTS chips.
We should probably break down nano-POTS for anyone outside the biology space. It stands for nanodroplet processing in one pot for trace samples.
Right, because the volumes are so small.
Exactly. When biologists work with a 10 micrometer cube of tissue, they are dealing with picoliters of fluid. If you drop that into a normal test tube, the sample evaporates or gets permanently stuck to the vast plastic walls. So nano-POTS chips are essentially microscopic glass slide test tubes designed to keep incredibly tiny liquid volumes contained and viable.
And the LCM samples were laser catapulted directly into those exact same nano-POTS chips. Both sets were then analyzed using an advanced mass spectrometry workflow called TIFF, paired with software called MS Fragger.
Finding the signal in the biological static.
Essentially, yes. The setup is designed to improve peptide identification in these low-input samples by heavily filtering out the chemical noise.
So the data is processed, the noise is filtered, and the results are overwhelmingly one-sided. The dicer utterly destroyed LCM in performance.
There wasn't even close.
Across all sizes, tested by 100 micrometers, 20 micrometers, and 10 micrometers, the 2pp printed dicer yielded significantly more unique proteins and peptides.
But if we connect this to the bigger picture, the most critical data points aren't just the overall wins. The massive paradigm shift happens precisely at the 10 and 20 micrometer spatial resolutions.
Because that is the exact scale where the photothermal heat from the UV laser and the glue-like adhesion of the PEN membrane utterly ruin the LCM samples.
Exactly. When you look at the mass spec data, the median log2 protein intensities at 10 micrometers for the NICER hit 17.4. The LCM method lagged behind at 15.1.
And since that's a log scale, we aren't talking about a marginal bump. That is a massive multiplier in the raw amount of usable protein signal recovered.
And the researchers also had to prove that the INSER wasn't somehow chemically altering the proteins differently than the laser. They analyzed the fraction of fully tryptic peptides, meaning the proteins were cut cleanly and properly by the digestive enzymes, and found they were similar between both methods.
They also looked at methionine oxidized peptides, right? Basically biological rust.
Yes. When a UV laser cooks a sample, it oxidizes the proteins. The data showed the dicer wasn't creating that biological rust. There is simply more physical, intact proteins surviving the mechanical shearing process.
We also need to highlight the reproducibility here, because if a pathologist is mapping a tumor ecosystem, consistent data is non-negotiable.
The coefficient of variation, or CV data, tells the whole story. At 10 micrometers, the dicer delivered a highly consistent median CV of 0.337.
And LCM.
The LCM method came in at a highly variable 0.505. The laser is just too chaotic at that microscopic scale.
Which brings us to the core takeaway for you, our listener. As you manipulate laser powers and push the limits of your 2PP systems, this deep dive proves that your field has evolved far beyond just manufacturing passive microoptics or standard microfluidic channels.
It's becoming foundational to biology.
Right. Two-photon polymerization is actively becoming the structural key to bypassing laser-induced thermal damage in clinical and biological research.
Because the multi-tiered dicer completely eliminates UV exposure on the delicate tissue. It abandons those problematic hydrophobic polymer membranes entirely. And it utilizes a single parallel mechanical pass to dice the tissue, completely avoiding the destructive friction of a macroscopic blade.
And none of this biological preservation is possible without the submicron resolution of 2PP printing, combined with that incredibly clever hierarchical geometric design.
It forces us to look at how we approach fundamental scientific roadblocks. We just saw how a completely mechanical 3D printed array can violently outperform a high-tech laser system simply by leveraging nanoscale geometry to dictate how force is distributed.
A 3D printed piece of polymer plastic beat a UV laser at its own game.
It really did. And this raises an important question, which leaves us with a highly provocative thought to mull over. If 2PP can precisely program mechanical stress distribution to physically outperform a laser at a 10 micrometer scale, what other traditional heat-dependent gold standard isolation techniques in chemistry or biology are just waiting to be disrupted by a clever 3D printed geometric structure?